All right, so week from today is exam 1. But before we do that, let's answer this question. What did the pen say to the pencil? What did the pen say to the pencil? So what's your point? All right, how about this one? Why do most fish live in salt water? Because they're allergic to pepper. That was her family name. I know exactly. That's her handwriting. Awesome. Pepper was crossed out. Oh, no. Oh, there, because she used the wrong there. She's still trying to figure it out. But she used the correct one after she lined it up. All right. Any questions before we get started? What's significant about this Thursday, 9/10 by 11:59 p.m.? Worksheet number 1 is due. So if you don't have any clue what we're talking about, you should probably come see me in my office hours today. Or come to SI section and ask them what the worksheets are. Because at this point, if you don't know about the Connect worksheets, you're going to be behind here very shortly, like in 2 days. And then Friday, September 11th, what's significant about that day with respect to this class? Quiz number 1 is due, okay, by 11:59 p.m. Now I'm getting a lot of questions like, I want to study, but I can't see my responses. Well, they will open up completely after the due date, and then you'll be able to see everything. So those of you that have already completed it, you can't see the whole quiz until Friday morning, essentially. Okay? But there should be plenty of time between when the quiz and the worksheet closes. Our quiz is Saturday morning, you'll see it. The worksheet, you'll see Friday morning. But there's plenty of time before the exam for you to check out what those answers are. Did I mention the copy reserve of the text? Yes. Okay, sweet. I have a note to myself. I thought I said it, but I couldn't remember. Some of you are like, I still don't know what you're talking about. So if you're missing the textbook, if you go to the free trial, do you have access to the ebook on that free trial? Everybody tried that? Yes. Yes. Okay. So that's one option. The other option is you can go bring your laptop to the library, log into your account, and you can check out a hard copy of the textbook out of the reserve desk. But you have to keep it within the library, and you have it for about 4 hours. Okay, so there's a copy of the textbook in the library for you to check out in case you're waiting for a Western Union from Mom. All right, this is where we're going to start. We ended on this dehydration synthesis, which is taking a monomer and adding it to another monomer and forming a dimer, which is 2. Out of that process, we squeeze out water. And if you go from a dimer in the reverse direction, if you drew an arrow in the reverse direction, you would actually call this hydrolysis or digestion. So what are some examples of carbohydrates? Again, we're in this section of the lecture about organic materials. Carbohydrates are some CHO to some repeating configuration to the end. They usually end verbally by name in os. They're hydrophilic, meaning they like water. If you think of sugar, like table sugar, and you had a glass of water, or you have tea, iced tea, or coffee, and you added in a tablespoon of sucrose, which is actually a type of a disaccharide because it's made up of 2 monomers known as glucose and fructose. You could spin around your spoon in that glass, and after a little while, you'll see it go into solution. Okay? If you're my children, it doesn't quite ever go into solution because you've added too many spoonfuls of sugar. Okay? And then it's just swirling around and it's saturated. Right? And apparently that's the best way to drink it. This is hydrophilic. That means that it likes water, so it goes into solution readily. We have some examples of monosaccharides like glucose, fructose, and galactose. And you can see sort of the nomenclature the organic nomenclature C6H12O6. Two main examples of disaccharides are going to be sucrose and lactose. Sucrose, shown up here, table sugar, is a glucose molecule, which is on the left, and the fructose molecule, which is on the right. So you can bind them together and make a disaccharide. Lactose is glucose on one side and galactose on the other. And then maltose is also another type of disaccharide, so you can add that to this list, or just draw an arrow, saying another example. And that's what? What are these two molecules? Glucose and glucose. So two glucose molecules forms a disaccharide known as maltose. And if you continue to add chains, you kind of roll out across this wall into the back and then over to here, you're going to form a complex carbohydrate known as a polysaccharide. And that would be specifically glucose strung together like that would be glycogen. So do you see how these chains are strung together? And they form a more complex carbohydrate. Some other examples of polysaccharides, starches, as well as cellulose. Now the complex carbohydrates, they take a little longer to break down or digest. Right? Hydrolysis or breaking of these bonds takes a little longer because they're all strung out in this long profile. And so if you have a meal full of complex carbohydrates, right, it'll last energy-wise a little bit longer, and you won't necessarily become hungry as quickly as if you have what we call fast sugar. So our fast sugars are going to be your monosaccharides or your disaccharides, like things like candy, things that are high in table sugar or glucose. It has a quick energy source, but it doesn't last very long because it doesn't take much time just to burn one particular glucose molecule. Make sense? So that's kind of the big difference that you see between complex sugars or polysaccharides and simple sugars like monosaccharides and disaccharides. Question in the back.
Well, I was just curious, if you're lactose intolerant, does that mean that you also have like a slight intolerance to glucose, or is it only like sugar, or is it only if it's put together to make lactose?
It's a good question. So the question in the back, if you can't hear it, is what happens if you're lactose intolerant? Okay, does that mean you can't handle lactose, or can you not handle glucose or other types of sugars? Well, so what's going on with a lactose intolerance is typically, not always, but most commonly, it's a deficiency, or you start turning off the production of a critical enzyme known as lactase. Lactase is an enzyme, it ends in ASE, and it's an enzyme that breaks down lactose. Right? Lactose is a type of sugar that you commonly find in milk and other dairy products. Well, we're one of the few organisms that continues to consume milk and dairy products outside of being infants. Okay? Kind of a little strange if you think about it this way, right? We're the only ones that continue to drink milk. And, even more weird, is it's from other organisms. Okay? Like, you think about that for a while, right? Not too long, because then your mind goes some places. Kind of weird. But, biologically speaking, what is the point of making lactase beyond infancy for most animals? It's not really necessary. And so that's probably one of the reasons that we see in our population this presence or this prevalence of lactose intolerance is we tend to turn off the gene for making lactase at a very young age. And in some individuals, it turns off completely, and they can't have it at all. It's extremely disruptive. In other individuals, it turns off and it kind of phases out as you get older. And so when you could have a nice big old milkshake when you're a kid, you might find later in life, like in your middle ages, you can't really handle that milkshake anymore. It gives you a stomachache. Okay? Does that answer your question? So it's really focused mostly on lactase.
Can you grow out of an intolerance though? I'm lactose intolerant. When I was younger, it was really hard for me to drink milk and things like that. But now that I'm older,
Do you take anything supplemental? No?
But like, the only thing I can't drink is like strictly milk, but I'm perfectly fine with any other kind.
So your body can adapt, and it will adapt. So, I mean, if you're continuing to consume something, and, you know, like you hear of people that have food allergies, and then they sort of grow out of their food allergies. What's happening there? We cover this more in my PATH class, but you can build up desensitization to an IgE, which is an antibody that you manufacture towards a certain allergen. It does happen. That's one of the reasons you see in a lot of naturopathic medicine, they encourage folks with bad allergies to actually consume local honey. Have you ever heard of this? So the local honey, these organisms, these bees, are gathering pollen from the local area. And so what you do is you start dosing yourself slowly over time. Okay, you don't like, right? I don't know, you might explode or something. You slowly over time build up your tolerance towards the honey and the natural pollens that are from the region. And that's why you don't want honey that's imported from other locations. You want natural honey from that particular area. But that's the whole rationale behind it. And it actually does work. And Allegra D also helps too, just in case you're curious. All right. Lipids. Moving out of sugars to lipids. They're hydrophobic. They don't like water. They avoid it. They have a carbon and an oxygen and a hydrogen with a very high hydrogen to oxygen ratio. They're less oxidized, which means they have a greater calorie per gram. In its format, on average, we can generate about 9.3 kilocalories of energy per gram of lipid. So you can see this picture here of this phospholipid. We have a nitrogen head, we have a phosphorus sort of backbone, then the oxygen, and then you have this carbon-hydrogen tail. Right? So the polar group, hydrophilic region, is the head is going to point towards the water or the aqueous environment. The tails are going to try to hide. So if you put a drop of oil or lipid in solution, it's going to form these little micelles. It's going to immediately organize with all the tails in the middle of the sphere and all the heads on the outer segment. And that's a little fat droplet that you see sitting in a glass of water. There are several different types of fat. Not all fat is the same. The first category, known as fatty acids, come in either a saturated or an unsaturated format. They're usually between 4 and 24 carbons in their backbone. And an unsaturated fat, the word saturation is referring to the amount of hydrogen that's bound to the carbon. Remember, that was one of our key definitions about organic, was a hydrogenated carbon. So in an unsaturated fat, you actually have open positions for more hydrogen to potentially bind to the carbon. So in other words, if you see a C double bonded to a C, then it's in an unsaturated format, right? Because if you broke that double bond, you can actually add more hydrogen atoms. A saturated fat is one that has as much hydrogen as it can carry. So this upper image up top is unsaturated. Why is it unsaturated? Because if you see pictorially this C double bond to that C and this C double bond to that C, if you cleave that double bonded carbon, you can add one more hydrogen here and one more hydrogen here, one more hydrogen here, one more hydrogen there. Make sense? So this would be an unsaturated lipid. If you look at the bottom example, this is cholesterol. Also unsaturated, because in this nomenclature, pictorially, you can see this double line represents a C double bonded to another carbon, right? So a carbon double bond right here. The cholesterol molecule also in a unsaturated format. Now, contrary to popular belief, cholesterol is actually good for you. It's actually important. It's critical for life. Now, the challenge here is in our diet, in our Western society, we actually get too much cholesterol in our diet. In fact, about 85% of the cholesterol that you need, you manufacture. You make it yourself. You only need about 15% supplemented out of your diet. I think that's equivalent to like 3 French fries at McDonald's. Okay.
Is there a name for the group that hangs out the carbon rings?
Yeah, so the group that hangs out the carbon rings, if you go back to our lecture on Thursday, are all named here on the right side of this graph. Does that make sense?
So it could be any of those.
So it could be any of these.
And that's
That's a
the difference between the different types of steroids.
difference between different types of steroids and different types of molecules, because if you look, you could actually have an amino group here, have an amino acid and a protein. You take that off, now you got nucleic acid if you add a phosphate group. If you strip that off and you just have an OH, now you're only in sugars or alcohols. Okay? Good question. So moving on. So the steroids are 4 rings. Cholesterol is this example of a sterol or a steroid. And we just went over that you manufacture most of what you need in your body, and you only need a little bit out of your diet. Now, if we move on to other types of lipids, we have what are called triglycerides, known as neutral fats. And this is a glycerol with 3 fatty acids. It comes in 2 different types of formats, oils and fats. What's the difference? Well, a room temperature versus the state of the material at room temperature is actually the main difference. So a fat versus an oil is a little arbitrary, but if it's a triglyceride that's liquid at room temperature, it's considered an oil. That would be like Wesson cooking oil or canola oil. These would be types of triglycerides that are in oil format. A fat would be something that is solid at room temperature. So the examples there would be like Crisco, right? Coconut oil, interestingly, is really a fat because at room temperature, it's actually in a solid format. Okay? Phospholipids. Big arrow over to the right. We've seen that on a previous slide. We have a neutral fat. We have a triglyceride plus an additional phosphate group. These are what we call amphiphilic. So amphiphilic here with a phospholipid means that it has 2 different characteristics. So on the exam, it would be incorrect to say that it was hydrophilic. It would be incorrect to say that it was hydrophobic. It would be most correct, and you'll see at the very top of the exam, it says, pick the most correct answer. So the most correct answer in a multiple choice question about a phospholipid next Tuesday would be which answer? Amphiphilic. So if you come and you want to argue, well, it's hydrophilic and hydrophobic, so I theoretically should have gotten either one of those right. I picked A, which was hydrophilic. But if there was a C answer that said amphiphilic, that would be the best, most correct answer to choose. Make sense? That is like a huge hint, just in case you're curious. Like if you're not catching really the hints, that's what I would highlight and write possible test question. Do I need to be more clear than that? That's pretty clear? Okay, sweet. Yes?
To go back to cholesterol, where in the body is that developed and how is it manufactured?
We'll go over how cholesterol is manufactured. We'll talk about skin. It's synthesized using the organ of the skin, but we'll get there. We'll build up. Okay, it's pretty cool. All right, well, this last family is called an eicosanoid. That's how you pronounce that, eicosanoid. If you were reading in the textbook and you came across that, you may or may not know exactly how to pronounce it. It's a 20-carbon compound. There's an example of prostaglandins to the bottom right. What in the world is an eicosanoid? This is a compound that's derived from the membrane of your cells. You take a cell membrane, you break it down, and you generate a base material known as arachidonic acid. And from arachidonic acid, you manufacture, your body makes, as the raw material, arachidonic acid, it makes a family of molecules known as eicosanoids. So we're in this fat section, a lipid section. So you take the phospholipid bilayer and you make stuff out of it. Okay? Part of the phospholipid bilayer has cholesterol within it. We're going to see that here in the next lecture. What do prostaglandins do? What are they good for? Well, prostaglandins are critically important for your defense mechanisms like with inflammation. They facilitate vasoconstriction of the blood vessels. And in some cases, they stimulate vasodilation, where you actually need more blood flow to come so you can get blood components out into the tissue in order to fight an invader. Here's some other types of eicosanoids. Eicosanoids. So things like prostacyclin, thromboxane. All of these are manufactured from the phospholipid bilayer. They assist in inflammation, blood clotting, hormone action. In some cases, they stimulate labor contractions in females. And like I said, they regulate the size of the blood vessel diameter. All of those families, 5 main families of lipids, yeah.
So do they act in the same way as hormones?
A lot of hormones are base molecule out of lipid. So, they're steroids. So the 2 most important ones in this room are going to be estrogen and testosterone, which are base steroid molecules, right? So they're sterols, made out of fat. Okay, so remember I was saying earlier in our lecture series that these sugars, these fats, these proteins, you need all of them in order to facilitate life. Now, if you have an excess storage of any one of them, then you could probably restrict it in your diet for a period of time, but you cannot eliminate it completely. You won't survive. Okay, I promise. Unless you're substituting in some other way. Okay, so the last little segment here are proteins and nucleic acids. We covered sugars, we covered lipids, and now we're going to cover proteins and nucleic acids. What are proteins? Well, they're made up of amino acids. Those are different than nucleic acids. Nucleic acids we'll get to. They make up DNA and RNA. The amino acids are what are used to make up proteins. There's 21 unique amino acids that are known to exist, and they could literally create thousands of different types of proteins, just from 21 different unique arrangements. Okay? But it shouldn't be too strange for you to think about how do you put 21 different things together and get thousands of proteins. We only have 26 letters in the alphabet. And think of the millions of words that we can actually generate using just 26 letters. So it's a similar type of alphabet. Does that make sense? That analogy, I think, is helpful. When you think about there's only 21 amino acids, why do we have thousands of proteins? You have millions or maybe even billions of words in the English language. Right? You guys are making up new ones every generation because technology creates all these new words. But there's still a 26-letter alphabet that actually defines them all. Alright, so amino and carboxylate group ends help to determine the groups. They also differ by their functional group, their R group, that's what that means. Of all of the amino acids, glycine is our simplest. They're often amphiphilic, meaning they have a hydrophobic and a hydrophilic region, kind of like lipids. Okay? And when we talk about digestion, right, which is chemical breakdown of food, we use certain enzymes in order to break down those materials. Well, the enzymes that we use to break down proteins are things like pepsin. Okay? They cleave certain peptide bonds. So, for example, if you look over here on the right, you're seeing one amino acid on the left and one amino acid on the right side of the screen. If you join them together, you're going to form a peptide bond and you make a dipeptide, putting 2 peptides together. When you eat a hamburger, right, you have large protein chains that, with mechanical breakdown of chewing, right, even some of the most impressive gentlemen in this class still have to at least chew 2 or 3 times to get the hamburger down into their tongue. Okay? So there's a little bit of physical, what we call mastication, or breakdown of that food, mechanically breaking it down. But then, to get the rest of that protein into its constitutive parts, we use enzymes to do that. And you're breaking these peptide bonds with things like pepsin. They're a chain of amino acids. Not sugars, they're a chain of amino acids. They're bound together by these peptide bonds, and we have a definition that we categorize based upon how long the chain is. So if it's less than 15 amino acids, we call it an oligopeptide. If it's greater than 15 but less than 100, we call it a polypeptide. And it really isn't defined as a protein unless it's longer than 100 amino acids in length. So when they're organized into these different structures. Much of this should be reviewed to you. Okay? But for sake of completeness, for exam 1 next week, I want to go over it. We have a primary sequence, which is shown up here, which is kind of like beads on a necklace, strung out. Okay? I don't know at Christmas time if you guys ever used to, take a needle and string and then put like popcorn or, different types of cereal through it and then use that to go around the tree. Have you ever seen that? So that would be like a primary sequence. That's how it would look. It's just like these amino acids on a string. Well, if you start folding these uniquely, you get into what we call a secondary protein structure. And you form things like an alpha helix or a beta pleated sheet that looks like a big piece of cardboard paper that you folded into like 12 different back and forth modes, and then it's accordioned out. Okay, these are beta pleated sheets right here. And then we also have a random sequence that it can fold into, where you've got certain functional groups. As you take this long string of amino acids, there may be an affinity from one side to another, and it forms kind of this U shape as it folds over. Okay, so that would be more like random organization. And then the tertiary level is where you get some additional folding and coiling of this protein architecture, and you get different functional groups seeing each other that never would have seen each other before. So the tertiary and then the quaternary protein structure is where we start really harnessing some unique functional characteristics of the protein. Because if you can imagine in your mind, you take a string of amino acids out, and then you kind of wind it into a coil, and then you have a pleated sheet, and then you have another coil. Now you have two coils that you could bring together. They would never have seen each other in that architecture unless they first coiled. Okay? So in our tertiary architecture is where we start seeing functionality of the proteins specializing. We get globular proteins and structural proteins that are a result of the tertiary architecture. And then quaternary is where you get two different polypeptides or more to see each other and sit next to each other. And now it's kind of like a team approach. Now you start getting massive functionality that never would have existed at the level of the primary architecture. So let's try so this example down here, to foreshadow, give you an idea of what to look forward to. This is actually the hemoglobin molecule. Okay? So the hemoglobin molecule is a quaternary architecture of two polypeptides. And one of them brings in two binding sites, and the other one brings in another two binding sites. Binding sites for what? Oxygen. And the oxygen specifically wants to bind to the iron molecule that's found right in the middle of the heme group. So you have 4 heme groups in hemoglobin, and right in the middle of the heme group is an iron molecule, and oxygen has a ridiculously crazy affinity for iron. And you know that because you've seen rust, right? So the iron component in metal, when exposed to oxygen, will bind the oxygen and oxidize it and turns it into a rust color. All right, so let's try your hand at this. Identify each level of protein structure. So what would this be right here? Secondary, very good. What's this one? Primary, very good. How about this one? Tertiary and quaternary. Wow, 100% A+. You guys are amazing. Well, not only do we fold them and can you memorize different shapes, we also have unique functions. So now we're starting to lay the groundwork for the more complex things that we're going to discuss. I know some of you are probably bored. I get it. But if we don't go over this carefully, there are going to be a couple of slides, even in today's lecture, that those of you that are bored are going to go, wait, what did you say? I didn't get the whole movement of ions thing. Can we go back? So we've got to go through it systematically. These proteins are critically important for all those functions on the right. Structure, like collagen. Collagen helps give structural architecture to your tissues, okay, as well as your bones even. Communication. Proteins assist with communicating from one cell to another. They actually bind. They link up via protein interactions, okay? And they also communicate with each other via these protein interactions, right? They tell each other what's going on. So one cell to another actually will communicate. They transport things through the membrane, where stuff will bind to the protein on the surface, and then it will be internalized into the cell. Proteins help to catalyze reactions because enzymes are actually made up of proteins. So they stimulate a reaction to take place. They help with recognition as well as protection. Recognition. So if I needed a new kidney and I surveyed 400 of you, because the real reason you're here is to serve Dr. Keller a kidney, right? And I look for a match. There wouldn't be a match in this room, even with 400 of you, that my body would take your secondary kidney and say, oh yeah, that's acceptable. It would reject it because your kidney would have your labels on it, your protein labels, and my body would recognize it was foreign. Okay? It's a little bit of a problem with a donor organ that we have to have a workaround. But it's actually really important for like the flu, or colds, or bacteria, or viruses, because you want your body to say, that's not me, and I'm going to kill it. Okay? Movement. Think at the cellular level. So the flagellum is made up of a complex organization of proteins. Right? The flagellum was the little tail apparatus on the end of the cell that would actually propel it through the water. You guys remember that? Cell adhesion. Well, our cells also move. If you take your and my skin cells and you plate them in a petri dish and you put them in an incubator, they will not stay where you left them. They will actually crawl across that petri dish. They also move via interactions between the cell and the bottom of the petri dish. So it's not just this flagella motor that you learned about in cell biology. It's also all the cells in our body. If you have a massive cut on your arm and cells were not able to move, you would still have a massive cut on your arm today. The fact that your wounds heal is evidence that cells move and they also adhere as they go, right? And you can actually, in time-lapse photography, take pictures of your wounds closing and prove that cells actually move at the macroscopic level. All right, so they go through conformational changes as well. What does that mean? That means they're going to change their shape. And there's a couple things that can do that. Number one is physical disruption. Okay? If you flick the protein, or you hit it, or you mechanically insult it, or stimulate it mechanically, it will actually change its shape. Okay? That's one way to do it. Another way would be with voltage. Mechanical stimulation is not on the list, by the way. So you want to add that. Voltage is number two. So, like your garage door, right? Probably half of you have never, I just push the button, it goes up. I don't know how it works. Well, it's actually, if the power goes out, what happens? Oh, dude, my car's in the garage and it's stuck. Why? Because electricity will stimulate the garage door to open. How many of you have been to like a gated community? And you drive up and you punch in the code, right, and the door opens. So those are all voltage stimulated gates. And that's a great thing to think about when we talk about voltage-gated channels. And we'll talk about those here in a moment. So electricity will cause them to change their shape and create an opening and then close. Or hormones can bind. As a ligand, it binds to the protein, has a binding site, and that causes a conformational change. And then the architecture of the protein is different. And that might trigger a response. Okay, so there's three main ways you can get conformational changes. Well, it's also really important to maintain proteins at the appropriate temperature and pH. So we talked in our last lecture about temperature and pH being under homeostatic regulation. So denaturing a protein means that it will become non-functional. And the two main ways to denature a protein is either by heat or by pH changes. So for example, what color is this egg before you turn up the heat? This right here, this white part. It's clear. What is it made of? You're like, yucky stuff. So you know that's what my 9-year-old said. Gross stuff on the body. Dad. Surprised it's not bloody. That's what she would say. What is it? It's protein. What protein is it? Albumin. Who said that? Nicely done. This is albumin. The white of an egg is albumin. And albumin is also found in your blood. It's the main component of your blood. So if you turned up the heat in your bloodstream too high, your blood would coagulate in the blood vessels. Okay? Another way to denature proteins is with pH. How many of you have ever heard of a dish called ceviche? What is it? Fish. And it's fish, which has protein, but you cook it using what? Usually citric acids, like lemons or lime juice or some marinade that has a very acidic pH, right? It's a very fancy dish. You pay a lot of money for this piece of fish that doesn't even, it looks raw, but it's actually denatured. Okay? So the next time you go and order a steak, you should say, I would like it lightly denatured if you want it rare. Okay? Or denatured plus if you want it medium. Okay? Denatured plus plus would be medium well. You with me? So these proteins have to be carefully treated in the body, otherwise they will be completely non-functional. What are some of the functionalities of these proteins? Well, they work as enzymes. They help to catalyze reactions. Like here's one over here, where we've got sucrose, table sugar, right, made up of glucose on the left and fructose on the right. Some of you are like, do I need to know that because it's colored like green and orange? No, I just knew that because it was down here labeled. Okay. So glucose on the left, fructose on the right, but it is a, what? Disaccharide, binds into this enzyme binding site, and that catalyzes the breakdown of those two enzymes. But if the pH or the temperature is out of whack, not under homeostatic regulation, it won't work. It will not work. Okay? And that's why fevers are such a problem in young children. And that's why when you're running a slight fever, you feel like crap, right? Because things aren't working correctly. You're lethargic, you're slow, don't have an appetite. Wounds actually heal slower. Okay? Metabolism is really, really focused on getting rid of the flu or the cold. And it's not being used for other types of activities. And that's why it's so hard to function when you have a cold. It helps catalyze reactions. The enzyme itself can be recycled and reused as long as it's not rendered non-functional or denatured. It also, as you remember back to chemistry, it helps to lower the activation energy. So in the presence of certain enzymes, the amount of energy required to run a reaction is lower. You're more efficient with the use of these enzymes. Okay? This is the enzymatic pathway that we just walked through. So lastly, in our carbon-based portion of the lecture, we need to talk about nucleotides. So nucleotides have a nitrogenous base, they have a phosphate group, and a sugar background, or backbone. And if you look at this adenosine triphosphate, our adenosine is our nitrogenous base, right, with nitrogen right here. And our ribose is our sugar backbone. And then we have 3 phosphate groups, right? You count them. Here's one phosphate group with the P, here's a second one in the middle, and here's a third one. And so adenosine triphosphate is also known as what? ATP. You guys remember ATP? Why is ATP important? It's the common currency for all cells from an energy standpoint. And ATP, every time you cleave one of these phosphate groups off, you liberate energy. Remember that? And so if you cleave one phosphate, it becomes adenosine diphosphate, ADP. If you cleave a second one and it only is left with one, it's called adenosine monophosphate, AMP. And then with AMP, all you have to do, right, the body's very green, all you have to do is add a phosphate, it becomes AMP, or ADP, add a third phosphate, and it comes back to ATP. It recycles. It's constantly recycled. They're very unstable phosphate bonds, and that's a good thing because it means it doesn't take a lot to break off one of those phosphate groups and liberate energy. It's manufactured during glucose oxidation. When you break down sugar. And what might be a frightening pathway, bring back cold sweats, right, is glucose oxidation. So, you have all these prerequisite courses, and they go over glucose oxidation, glycolysis, and then aerobic respiration. And by now, you've had it like 12 times in your career, right? You're like, why do we keep going over this crap? I couldn't remember it the other 11 times. Okay? Well, you got to get it, guys. And what hopefully you're understanding is, when we keep repeating things in classes, it's because it's critically important. So, I'm not going to hold you to the fire on all the intermediates. But I do want you to understand some of the basic take-home principles of this old material. It should be old to you because of prerequisite coursework. So glycolysis, the pathway of glycolysis is where you take a sugar and you split it, you break it down, you digest it into 2 pyruvic acid molecules. And in the process of doing that, you liberate 2 ATP. You have a net of 2. Right? So you can see right here, here's glucose going through glycolysis. You could write glycolysis and draw an arrow, that's what this is. You generate pyruvic acid and you kick out 2 adenosine triphosphates that can be used as an energy source. If you don't have any oxygen available, let's say it's an exercise activity that's anaerobic, that means without oxygen, like a sprint. Okay? Like a 10-second sprint. Well, you're going to only create ATP. This pyruvic acid will convert to lactic acid. And that's actually that little burning sensation that you start feeling in the tissue. And your body clears it really quickly. It is acid, yes. Isn't that dangerous? Yes. But, good news, your body buffers it fast. And you just start doing this. Right? You breathe out. In fact, you even breathe heavy after you're done running because you're still blowing off that acid that you created. Well, if you have oxygen available, then you shuttle this to the mitochondria, and this is where the fun begins, right? This is where all that memorization took place. Like, oh crap. This is considered aerobic respiration because now it's in the presence of oxygen. And, of course, here, what we call this aerobic respiration, we also refer to it as what? Krebs cycle, tricarboxylic acid cycle. What's that? citric acid cycle is like 3 different names for the same thing, right? Because all these people had to, work on it simultaneously, and then they couldn't agree on calling it something, right? Well, eventually, oxygen accepts the final electron, but it allows the most amount of energy to be liberated. So in the presence of oxygen, you're going to get 36 additional ATP. So if you took one glucose molecule all the way through glycolysis and aerobic respiration, which includes the Krebs cycle or the tricarboxylic acid cycle or the citric acid cycle, plus the electron transport chain at the end, that all together gives you 36. So 36 plus 2 is how many? I know this is a bio class, it's not engineering, but can we use your phones? 38 net ATP. That is the correct answer on the exam. Okay? What are some other nucleotides other than ATP? ATP is the one that we're going to spend the most time on this semester. Because you have to have ATP to contract muscles, right? In fact, if you don't have ATP available, we call this something very specific that's found in many murder mystery movies. What's it called?
DNA.
What's that? DNA? No, if you don't have ATP, what happens to the body with no ATP being manufactured anymore? It's dead, and you call the complex what? Anybody know? Rigor mortis. Rigor mortis is when the muscles are basically locked in place because there's no ATP left. The person's been dead. We'll talk about it more this semester. But that is pure evidence that ATP is necessary for muscle contraction as well as muscle relaxation. But here's some other ones that are important. Maybe that's where the DNA came from. The shout out was just hurry up and finish the slide. Okay? Other nucleotides. Adenosine triphosphate, cyclic adenosine monophosphate, as well as our 2 critically important nucleic acids, DNA and RNA. In this picture here, what is this? That's a DNA strand example. Okay? So in this lecture we talked about inorganic and we talked about organic. And one of the big take-homes was both of those are critically important for life. Question?
Yes, I was just wondering, so for those 2 organic requires ATP for muscle contraction, so does that mean there's like leftover ATP for the Labyrinth effect?
Yeah, there's going to be ATP remaining in the body for a period of time. The Rigor complex doesn't really set up until about 6 hours post-mortem. Right? It peaks at about 24, and then it kind of fades over. As soon as the proteins start degrading or decaying, spoiling, then it doesn't matter if you have ATP, they just disintegrate apart from each other, and then the body's flaccid again. Okay, movable.